Gabriel. I'm going to present Module 3 of Chapter 1 to you, which is all about the scientific method. After reading the module, you should be able to explain all the details of the scientific method, and more importantly, you should be able to write your own experiment using the important criteria that you're going to learn about here and recognize things that might be wrong with an experiment that's not well designed. We're going to study the process and methods of science by applying this module to a classroom lab where we investigated seeds. We might not be done in the lab yet, but that's okay. You'll still be able to make those connections. While taking notes, I would recommend you create text notes in one color and classroom lab experiences in a complementary color. So you should have your notebook open, and the title in your notebook should be for a new page, Chapter 1, Module 3. You should open up your textbook, and you should look at the pages for Module 3. And in the title on the top of your page, you should write all the pages that are included in Module 3. Remember, each module should take up one full page in your notebook. Feel free to pause if you need to. These are the basic steps in a scientific investigation. You've learned these throughout elementary and middle school, but just in case you napped through it all those years, here it is again. So I have in white the basic steps, and in yellow I have how it applies to the seed lab. So we observe and we question. For the seed lab, the question was, does salt affect seed germination? Then we form a hypothesis from our question or make some type of prediction. And I recommend for this course and this year in your SMA life that you form a testable hypothesis using the if-then rule, which I'll go over in a lot of detail as this slideshow goes on. Then we collect data, conduct the experiment, which was the seed test that we set up. So I gave you a well-designed experiment, and you're going to see how all of the principles of a well-designed experiment have been used in that experiment. Then, we probably haven't gotten to this yet, we interpret the results by making graphs, conducting statistical tests, and then we disseminate our findings by drawing conclusions and reporting this to others. Sometimes we investigate and come up with new hypotheses based on our observation. Maybe you thought the seeds with no salt water will have the highest germination rate. That's fine if you did, but what if you observe conflicting observations? Well, then you have to reject the hypothesis and develop a new one. That's okay. It happens all the time when we really don't know the answers. It's how we learn, which is why we have this little arrow here. So we might have a hypothesis, collect all this data, and realize, oh, well, this isn't what we thought we were going to find, and then we come up with a new hypothesis, and we test again until we don't have to support our hypothesis, we just have to keep asking questions. That's real science. When writing a hypothesis, it must be testable. A testable hypothesis is easier to write if you use the if-then rule. Write down the if-then tips that I have written here for you. I is for if, which is for the independent, starts with the letter I, variable. Then is for the dependent variable. That's the one you don't manipulate. You can't just write anything in your if and then. They must be testable and measurable. And how do you know if they're testable or measurable? Imagine your if and then in a graph. Imagine your if on the x-axis and your then on the y-axis. For example, the effect of walking speed on heart rate. Think about that for a minute. These are very measurable. How do we know they are? They have units. Heart rate is in beats per minute. Walking is on speed, like miles per hour. So try writing a hypothesis using these little tips I gave you for the effect of walking speed on heart rate. You need the if and the then to have the measurement part made clear. Write a hypothesis for it. Go ahead and try. Hit pause and write a hypothesis. Did you write something like this? If walking speed increases, then heart rate increases. If not, don't get worried. Write down the parts you had missing. Notice that I can quickly imagine these variables on a graph. That helps. Imagine walking speed on an x-axis and increasing. What's that unit you're going to use? Maybe miles per hour? Imagine the dependent variable on the y-axis, also increasing. Draw the graph to help you remember. Draw the graph so that you remember if is the independent variable, and you have to have units. Y is the dependent variable, and you have to have units. They both have to be measurable. It has to be testable. All right, now try writing another hypothesis. Write one for the effect of sleep on grades. And remember the tips that I gave you. Pause the video until you're ready, and here's a hint. If you can visualize the graph and plotting your data on it, then you are probably writing a testable and measurable hypothesis. Oops. Okay, there it is. If the number of hours of sleep per night increases, then grade percentages will increase. Of course, experiments can test in more advanced ways with the use of fancy stats, but we're not there yet. So how did you do? All right, write down whatever you may have missed. Reflect on how you could have made it more measurable, and let's try another one. Write a hypothesis using units, variables, direction on a graph like increase or decrease to determine if salt water affects seed germination. Okay, pause here. Does salt water affect seed germination? How would you write it in an if-then statement, making sure you have units, variables, and direction? How'd you do? This is how I would have written it. You didn't have to write it my way. If the concentration of salt in water increases, then the number of seeds germinating decreases. So, I have concentration, that's my unit, that would be in percent. And that's my variable, and that's my direction. Then I have the number of seeds, that's just a count, that's my unit. And germination of seeds is my variable, and decreases is my direction. Okay, now if you're struggling with writing that alternate hypothesis, you can always start with the null. A null is as simple as there's just no effect of one or the other. There's no relationship. Start there, and then you can identify your variables more easily. They still have to be measurable, though, even if there's a null. They have to be measurable, and they have to have units. So when you design an experiment, you should consider the following criteria, and these all apply really well to our seed lab. We performed replication by having several groups repeat the same experiment. Several groups of students conducted the same exact lab experiment. So we had a lot more data, and in addition to that, the lab was replicated again and again and again. So whatever results one group got, it's better when more groups can replicate the same results with the same experiment. Same number of seeds, same saltwater concentration, same way of observing, collecting, recording data. We also used 10 seeds per concentration in order to have a larger sample size. Our sample size is N. So we had a larger N because we didn't even use just 10, because you really look at the whole class, we could have had over 100 seeds in a sample size for one concentration. The larger the sample size, the better. If we only used 1 seed per concentration, and it was a bad seed, we may have drawn inaccurate results. For accuracy, that was measured by collecting our data carefully, using extra eyes and a magnifier to zoom in this case. We increased our precision by getting similar results among most of our groups. That means it was a well-designed experiment. However, there was some uncertainty in our experiment because the seeds could have been bad seeds and not viable, rather than just being killed by the salt water. And we could have assumed that they didn't germinate because of salt water, where it may have been because they were sitting in a really, really dry, cold, or warm closet for the summer. Now, if you're struggling with the difference between accuracy and precision, I would draw this to help you understand it a little better. So think of accuracy as the bullseye here. If we were accurate, everything would hit the bullseye. So we have high accuracy over here. We're closer, everything is closer to the bullseye. But we had high precision because this may not have been a bullseye, but wow, everybody seemed to hit their target in the same place. So that's higher precision. And then in the second one, we had a lower precision. They were hitting all over the place, but they were all closer to the bullseye. Then we interpret our results. This is still another piece of the process. So we analyze the data, and then we make reasonable statements about the data. They can be inductive or deductive. You can read this all on your own. And then we decide, do we want to reject our hypothesis or accept our hypothesis? Reject means, yeah, that's not what I thought. Accept means, wow, this is exactly what I thought we were going to find. And either is fine. And then scientists have to disseminate their findings. So in middle school and high school labs, you typically disseminate your findings by writing a lab report, drawing your conclusions, and discussing it. In the real research world, you're publishing a paper to share your findings, and then others are learning from that. And if everybody else gets the same results by looking at the same things you did, the hypothesis becomes a theory. So with the seed lab, we are conducting a controlled experiment. It's in a classroom. All groups of seeds have the same exact conditions. All of them are in petri dishes. Then they're all put in a bag, and they're all put in the same location. All seeds were from the same original source. All seeds got the same amount of solution. All seeds were given the same amount of time to germinate. This is a very well-controlled study. Feel free to pause and review this part in your textbook. It's a nice example of a well-designed and well-controlled experiment using a lot of the things that we just discussed in this show. So many environmental research projects take place in the natural environment. So we just talked about everything in the laboratory. That's very easy to control, and we can manipulate everything very easily. But in the natural environment, that's a whole different story. These are typically not as well controlled and take sometimes many years or decades to collect the data and analyze it. Natural experiments are also not manipulated by the researcher. We don't manipulate the if, the independent variable. The if is not manipulated. The if just happens, and the then just happens. Examples of natural experiments can include the effect of seawater temperature on hurricane strength. Studying hurricanes is a natural event, and the location always differs. Another example could be the effect of earthquakes on landslides. We don't make the earthquake, and we don't make the landslide occur. We just find where these events occurred and make observations and collect data and look for correlations. Environmental science research is challenging because we have no control group. We only have one Earth, plus all these other challenges that are listed here. But studying, observing, and questioning what we know will help us understand how to be more sustainable and more Earth-friendly and help the human population survive on the only home that we have, Earth. This is the end of Chapter 1, Module 3. Please go back over the notes to make sure you can identify components of an experiment, and so that you can design one of your own experiments using the critical components required for this course under the appropriate scientific method principles, and think about scenarios that I could have given you. You should be able to recognize what the independent variable is, what the dependent variable is. Write a hypothesis for an investigation that you're given. Go over the review questions, go over the end of chapter questions, go back on your notes, highlight things, put boxes around things, color code things. That's the end of chapter 1. You have a quiz coming up soon. This is Mrs. Gabriel.